Hi, my name is Justina Jong Lee Sim and my metric number is 99365.
Hi, my name is Christine. My number matrix is 98519 and I'll be talking about the conclusion part of this video.
Hello guys, my name is Hisambi Muhammad Hasli and my metric number is 98920.
My name is Brian Som, my metric number is 98370, and in this video I'll talk about what our group has learned and the challenges that we face in this experiment.
Hi, my name is Nabila Mushida and my metric number is 100033.
This experiment is involving the usage of spectrophotometer in protein analysis. The topic for this practical one is spectrophotometry. There are three main objectives in this experiment. First of all, this experiment aims to introduce the students on the function of spectrophotometer in qualitative and quantitative of a biological molecule. Next, this experiment is ran to determine the concentration of unknown protein solutions A, B, and C. The last objective of this practical is to calculate the absorbance of the BSA protein standard at 595 nanometers. There are four materials that are used in these practicals. The material used include distilled water, Bradford reagent, 1 mg per ml BSA protein and standard unknown protein solutions A, B, and C. The apparatus that are used in this practical are test tube and test tube rate, micropipette, cuvettes, spectrophotometer, and vortex mixer.
First of all, we prepare 7 test tubes. 6 of these test tubes were used for our protein samples, while the remaining test tube was used as the blend solution. We made sure to label each tube clearly to avoid any confusion later on in the experiment. Next, we added 4 ml of distilled water to each test tube. To ensure precise measurement, we used a micropipette to accurately measure the water. This was essential to maintain consistency across all the test tubes. Additionally, we added 1 ml of Bradford reagent into each test tube. The Bradford reagent contained a dye that binds to the protein, causing a color change. This change in color is what we will use to measure the protein concentration in the sample later. Then we added 50 microliters of BSA protein standard into the second and third test tube. After that, we added 100 microliters of BSA into the fourth and fifth test tube. Finally, we added 200 microliters of BSA into the sixth and seventh test tube. But we left the first test tube as the blind. It only contained distilled water and Bradford reagent without any BSA protein added. Afterwards, we mixed all the test tubes using a bench mixer. This step was crucial to ensure that the Bradford reagent and the protein were well mixed and the color change would be uniform across all the tubes. Then we transferred the solution from each test tube into individual cuvettes for spectrometer analysis. Following that, we measured the absorbance of each test tube at 595 nanometer using a spectrophotometer. First, we placed the blend solution in the spectrophotometer and zeroed the machine. This was done to ensure that any absorbance reading taken from the samples could be accurately adjusted for the baseline provided by the blend solution. For the part 2, the method was the same as in part 1. But this time we used the unknown protein sample A, B, and C. We followed the same step for prepare and mix the solution. Before taking any reading, we used the blend solution to zero the spectrophotometer. After that, we measured the absorbance of the sample A, B, and C at 595 nanometers. Finally, we recorded the absorbance value for all 3 samples.
According to the conclusion, we are going to start with the standard curve establishment. On the left is the tabulated data of the results and calculations, and on the right is the graph generated from the table on the left. It's confirmed that the absorbance of the sample at 595 nanometers is directly proportional to the concentration of protein. As we can see from the graph of the absorbance increase with the increase in the volume of bovine serum albumin protein, otherwise known as BSA protein. The directly proportional graph demonstrated in the graph also validates the Beer-Lambert law. Next is the quantification of unknown protein. When deriving the graph of the standard, it gives the equation y equals to 0.02x. And using this equation, we can calculate the concentration of the unknown protein here, with y representing the absorbance and x representing the concentration. This experiment is aligned with the objective by familiarizing with spectroscopy techniques mainly knowing what to do and avoid to finally get an accurate reading. Next is by accurately calculating the concentration of the unknown protein and calculating the absorbance value of BSA proteins at 595 nanometer at different concentrations of the BSA protein. And from there, we managed to establish the relationship between the absorbance and the concentration of protein. Any significant observations other than previously discussed are that the Bradford technique allows for efficient analysis of protein concentration. The replication of data by having more than one result ensures data reliability and accuracy, and it can be seen from the graph previously shown that the standard deviation is equal to or less than 0.01. And finally, proper calibration techniques can ensure minimal error and high accuracy of the absorbance readings.
One of the primary learning outcomes was understanding the fundamental principles behind spectrometry protein identification and quantification. This includes mastering the concepts such as basic laws of light absorption and Beer-Lambert law to determine the absorbance and concentration values of our samples. We also learned how to handle the instrumentation of the analytical tool used in this experiment, which is the UV-Vis spectrometer, to understand the principles behind how the tool worked and what it could analyze. We also learned about the suitability of measuring apparatus use. In this experiment, we were given 3 sizes of pipette, each with a different range. We had learned which pipette to use depending on the volume of our samples, how to calibrate them to the desired volume, and the proper tip size used. One of the challenges that we faced during this experiment was sample preparation, which plays a crucial role in the reliability and reproducibility of results. During the experiment, we had forgotten to switch to a new pipette tip when handling the different samples. We were quick to realize the contamination and promptly redid the experiment with new samples and make sure to switch out the pipette tip after each use. Overall, this experiment has significantly enhanced our understanding of protein analysis via spectrometry. Despite the technical and analytical challenges, the opportunity to apply these methods firsthand has been valuable for developing both practical skills and scientific insight.
Here are some suggestions to improve the experiment in future sessions. Firstly, ensure the spectrophotometer is calibrated with a fresh blend sample before each measurement to maintain accuracy. Second, always use a new pipette tip from each sample to prevent contamination and ensure precise measurements. Third, check for air bubbles in cuvette before measuring absorbance, as then can interfere with light transmission. Fourth, handle cuvette by the top or edges to avoid contamination and maintain accuracy. Lastly, perform measurement in duplicate or triplicate to improve reliability and reduce errors. By following this step, we can ensure more consistent and accurate results.